In mammals, different Hox genes are expressed beginning at the hindbrain and through the tail region of the embryo. This expression pattern, as pointed out previously, is based on the gradients of retinoic acid, Wnt3A, and FGF8. As with Drosophila, the expression of the Hox genes along the anterior-posterior axis of the embryo corresponds to their 3 to 5' location on the chromosomes. So genes that are located in regions that are closer to the 3' ends of chromosomes are expressed more anteriorly, and those that are located in regions closer to the 5' ends are expressed more posteriorly. A difference between Drosophila and mammals is that in mammals there are up to 4 copies or 4 paralogs of each of the Hox genes, whereas Drosophila only has 1 copy of each Hox gene. Bear in mind, when I say 4 copies or up to 4 copies, what I mean is up to 4 sets of each Hox gene, because we have to consider that there are both maternal and paternal alleles. So this means that there would be a maternal and paternal copy of each of the Hox genes. So for some of these Hox genes, the ones that have 4 paralogs, there would be 8 copies in total if we consider both the maternal and the paternal copies. Similar Hox genes in which are located in the same region of the chromosome are considered or classified as paralogs because these are thought to have formed by chromosome duplication events. There are 13 Hox genes in mammals, which are numbered, as you can see here, from 1 through 13. And the paralogs are denoted by letters that range from A to D. The 3' end located Hox genes are not only expressed more anteriorly, but they're also expressed earlier in development. On the other hand, the Hox genes that are located in more 5' regions of the chromosome are expressed in more posterior structures, and they're expressed later in development. That temporal and spatial pattern of expression is thought to be associated to changes in chromatin structure. The chromatin for Hox genes that are located at the 3' locations of the chromosome become euchromatic earlier in development compared to those Hox genes that are found closer to the 5' end of the chromosome. And this is illustrated in this figure. As you can see, the inactive Hox genes are shown in red and the active ones are shown in blue. So you can see that earlier in development, the more 3' located Hox genes become activated. And as development progresses, we have the gradual activation of the genes beginning at the 3' end and moving to the 5' end. So what is the result of the expression of these different Hox genes? Well, as in Drosophila, they give each segment of the mammalian embryo its unique identity. Hox genes are expressed in the somites, which give rise to the vertebra and also to the muscles of the back. And the Hox genes are also expressed in other tissues like the nervous system, the reproductive system, and the neural crest cells. And we'll discuss these tissues and the effect of Hox genes in these tissues later in the semester. For the time being, we're going to use vertebra as an example to illustrate how Hox genes regulate segmentation and thus patterning in the embryo. If you look at the skeletons of the chick and the mouse, which are shown here, you can see that the chick has more cervical vertebra and less thoracic vertebra, and the mouse has more thoracic vertebra and less cervical vertebra. This diagram represents the different types of vertebra in the chick and mouse, along with the patterns of expression of the different Hox genes. As you can see, the chick has 14 cervical vertebrae while the mouse has 7. What this figure shows is that the expression of the different Hox genes is correlated to the type of vertebra more than to the location of the vertebra along the anterior-posterior axis. In other words, a particular Hox gene determines the type of vertebra that will form at a given location. For example, the HOX5 gene is responsible for the formation of the cervical vertebra. As you can see, this gene is expressed along a wider region in the chick compared to the mouse. A very interesting thing about Hox genes is that once a somite, and remember that's what's going to give rise to the vertebra, once those somites begin to express a particular Hox gene, they will retain their expression. This means that if we were to move a somite that gives rise to a thoracic vertebra, Once that soma began to express the Hox genes that are responsible for specifying the thoracic vertebra, which are HOX6 and HOX9, if we move that from an older embryo to a younger embryo, these somites would still give rise to thoracic vertebra at their new location. And this is an example in the chick, but the same principle applies to mammals. Alternatively, if the expression patterns of Hox genes are altered, this can result in the transformation of one type of vertebra to a different type. So these homeotic transformations are very similar to what we saw in Drosophila. For example, the Hox10 paralogs are responsible for the formation of the lumbar vertebrae. This is in the wild type animal. When the Hox10 paralogs are deleted, which is shown here in part B, you can see that the lumbar vertebra resemble the thoracic vertebra. So deleting a Hox gene leads to the duplication of the anterior segment. Similarly, when Hox11 paralogs are removed, which is shown here in part C, the sacral vertebra develop into lumbar vertebra instead. The mechanism for the specification of the left-right axis in mammals is very similar to the mechanism of amphibians and birds. Although the figure at the bottom here displays the mechanism of left-right axis specification in avian embryos, the process is similar in mammals, so we're going to use the figure to discuss the process. As you can see, the Perkin factor nodal is expressed in the left side of the embryo, where it activates the expression of the transcription factor PITX2. This transcription factor is important for the left-right asymmetry of organs. On the right side, there are inhibitors that prevent nodal from being expressed. The asymmetric localization of nodal and its inhibitor is the result of the movement of cilia that are located in the node. This process involves single motile cilia on node cells, which create a current. Then single non-motile cilia on cells that surround the node sense the current, and they respond by suppressing the synthesis of the nodal inhibitor Cerberus. So the result of this is the asymmetric distribution of nodal and its inhibitor Cerberus on the left and right-hand sides of the embryo. The last topic we're going to be discussing as it pertains to mammalian development is twinning. Because human embryos exhibit conditional specification such that cells can compensate for missing cells, two or more organisms can develop from the same fertilized egg as a result of the separation of either totipotent or pluripotent cells in the early embryo. It's important to know that the twinning events that we're going to discuss refer to identical or monozygotic twins, which are derived from a single fertilization event, in other words, the fusion of one egg and one sperm. Dizygotic twins are different than this because they result from two separate fertilization events, in other words, two eggs, each of which are fertilized by a different sperm. The majority of monozygotic twins, approximately 65%, share the same placenta, but each twin has its own amnion. About a third of monozygotic twins each have both their own amnion and their own placenta. And in a few cases, monozygotic twins share both the placenta and the amnion. Whether or not twins share a placenta and/or an amnion depends on when the separation event took place during development. If the separation event happened very early before the differentiation of the trophectoderm cells, each twin will have their own placenta and their own amnion. If the separation event happened after the formation of the trophectoderm, but before the separation of the tissues that give rise to the amnion, then the embryos will share the placenta, since the trophectoderm had already formed, but they will each have their own amnion. Finally, if the separation event occurs after the formation of both the trophectoderm and the amnion, then the twins will share both the placenta and the amnion. Whereas monozygotic twins are formed as a result of an event that occurs during cleavage, conjoined twins are thought to be the result of defects that occur during gastrulation. It's not well understood why this happens in humans, but studies in other organisms like the chick suggests that the formation of multiple axes of gastrulation and the fusion of primitive streaks may be responsible for conjoined twin formation.